Hello, everyone, and welcome to today's live broadcast, Commercializing Nanomedicine for Improving Human Health. Not just a hype, it's really happening. Presented by Dr. Thomas Webster, Professor and Department Chair of Chemical Engineering, and Art Zafiropoulos, Chair in Engineering, affiliated faculty in Bioengineering at Northeastern University College of Engineering. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that if you have any questions, please click on the Ask a Question box located on the far left of your screen. Type your questions into the drop-down box appearing on that screen, and our presenter will address your questions with answers via email after the presentation. If you have any trouble seeing or hearing this presentation, just click on that Help Desk button located on the promotional board at the bottom center of your screen, or use that Ask a Question box and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the accreditation box button located at the promotional board at the bottom center of your screen and follow the process for obtaining those credits. Without further ado, please join me in welcoming Dr. Thomas Webster. I will now turn the presentation over to him. Welcome, sir.
Very much for that nice introduction and the opportunity for me to present some of the groundbreaking research we are doing in the area of nanotechnology. So I have about 20 years of experience looking at how the body interacts with nanomaterials. And what we're finding out, and what you'll hear in this presentation, is that there are very controlled ways to dictate what the body does using nanomaterials. And the most exciting thing that I'll present to you is that we are able to control body functions using nanomaterials without pharmaceutical agents. And I'll describe why that is such an important objective for us in this presentation. So first of all, the reason why we are focused on nanomaterials and not pharmaceutical agents to combat different disease is something called the emergence of antibiotic-resistant bacteria. While I think we all know that antibiotic-resistant bacteria is a huge problem, we may not appreciate just how large of a problem this is going to be in the years ahead. In fact, many different thought agencies, U.S. government agencies, as well as worldwide agencies have recognized the problem that we will face with antibiotic-resistant bacteria in the years ahead. On this slide, you can clearly see some statistics that places like the Centers for Disease Control are reporting. This graph that's indicated here indicates that by the year 2050, more people, in fact, an estimated 10 million people, will actually die from antibiotic-resistant bacteria, antibiotic-resistant infections. This is alarming when you consider how this compares to other diseases that we're used to seeing such harmful statistics for. So also pictured on this slide is an estimated number of people that will die from cancer by 2050. And it's roughly 8.2 million is what the Centers for Disease Control, or the CDC, predicts. So really what this statistic should say to us is that more people are predicted to die from antibiotic-resistant bacteria infections than all cancers combined by 2050. This is amazing and just thought-provoking to think about, because if you consider all of the resources that we have put towards fighting cancer, the amount of money, and I would say the amount of energy that we have put towards fighting antibiotic-resistant bacteria pales in comparison. Now, of course, we want to reduce cancer even more than what we're doing today, but we also need to pay attention to strategies that can reduce the emergence of antibiotic-resistant bacteria. Now, these statistics should not only be alarming to us, but they should also highlight that perhaps our approach to use antibiotics to kill bacteria is not working. And as we know, there are many fields that have emerged. In particular, nanotechnology is one that I'm pretty excited about, that we are looking at fighting bacteria without drugs to keep these statistics from occurring, to keep the growing number of antibiotic-resistant bacteria from growing, which is affecting the world population. So what I'd like to do in the very beginning here of the presentation, after highlighting how important it is for us to fight antibiotic-resistant bacteria, I would like to highlight some fundamental definitions in nanotechnology, since the average person might not know that much about nanotechnology, and quite honestly, it's a relatively new field that is very broad, but yet, again, very exciting. There are many reasons to look at nanotechnology to fight bacteria without drugs. One of the first things you can do is simply look at nature. Look all around at living organisms in nature, and you find beautiful, very distinct nanoscale features. This is just one of many examples that I could provide. If you consider a cicada or a locust or any kind of insect, and you look at the wing of that insect, you will find many distinct nanoscale features. In fact, that's what's pictured in B, is a high-resolution microscopy image of what the wing of an insect looks like. So why is this important? This is important because if you try to grow bacteria on this insect wing, they don't grow. Whether it's a gram-positive bacteria, a gram-negative bacteria, an antibiotic-resistant bacteria, you cannot get them to grow on these kinds of surfaces. So compare that to the kind of surfaces that we implant on our medical devices today. They don't possess nanoscale features. So the hip implant that you might have or your grandparent might have, the vascular stent that somebody might have, the catheter that might have been used to help deliver a blood transfusion, they don't have nanoscale features on their surface. They are what we call nanosmooth. So it begs the question, if nature has evolved to have these very unique nanoscale features, why aren't we using them in everyday life to reduce bacteria growth, to reduce infection, to really reduce the emergence of antibiotic-resistant bacteria? And that's one of the things we have been focusing on. So if you, again, consider what this means for a medical device, what this means for medicine, what I have included on this slide, these two pictures, is what the surface of an everyday hip implant looks like. And I've labeled that today's implant. As we know in many hip implants, in many orthopedic applications, those micron features that you see in today's implant actually improves bone growth. Unfortunately, have been shown to increase bacteria growth. Bacteria are about 1 micron in diameter, most of them, and they can easily get stuck. They can easily situate in these larger micron features on today's implant. In contrast, what I've also included on this slide is a nanostructured implant. And there are many ways that you can transform today's implant into a nanostructured implant. But if you have nanoscale features on that implant, those features are too small to allow for that micron-featured bacteria to situate inside that material. So there are many, again, exciting opportunities and exciting reasons to look at nanostructured materials in medicine. They mimic nature, and nature has been able to reduce bacteria growth on some of these surfaces, and they also have size dimensions that keep these micron-structured microorganisms like bacteria from attaching to them. Many people actually refer to these nanostructures as a bed of nails. And these bed of nails, again, while they can allow for tissue to form and they can allow for larger mammalian cells to attach. They do not allow for nanostructured microorganisms, I should say, like bacteria, to attach. So while this argument of biologically inspired features mimicking the wing of an insect sounds great, we've also been interested in a more deeper explanation for why bacteria don't like these surfaces. And I'll show you some of the surfaces that we've created, telling you how we've created them, that reduce bacteria growth. But I want to talk about this, of why bacteria don't like nanostructured features, whether it's an insect wing or something we put on an implant like a catheter or hip implant. And really it has to do with something called surface energy. So every surface has energy associated with it. And what basically surface energy means is how hydrophilic or how hydrophobic something is. How well does a water drop spread on a surface? That's a measure of its energy. When you take a sample, an implant, an insect wing, and you have nanoscale features on that wing, you change energy of that surface. So what I've pictured on this slide, what I have diagrammed on this slide on the left in green, is a surface. Think of any surface that has nanoscale features. It changes in energy quite simply because you're changing surface area. So if you envision a flat surface that's made of titanium, titanium is one of the most popular orthopedic implant chemistries, it will have a certain surface energy. If you then take that titanium and put these nanoscale features on there, like what's diagrammed in green, you have now increased the amount of titanium that's exposed. That increased amount of titanium, increased surface area of titanium, leads to different surface energy. More exposure of a hydrophilic chemistry will give you more hydrophilicity. Similarly, if we started with a hydrophobic chemistry, like a polymer, and that polymer, we consider what its energy is when flat versus when it has nanoscale features, we quickly can understand that there's more surface area in that polymer when it has nanoscale features, and that in turn can lead to more hydrophobic interactions because you have more hydrophobic polymer chemistries exposed. So why is that important, that we have changed the surface energy of a material when putting nanoscale features on it? The reason why that's important is in our bodies, we have countless proteins that will initially adsorb to any medical device, any material you put in the body. You'll get immediate adsorption of proteins. Every one of those proteins has different energy. So if you start changing the energy of your surface, you're going to change what proteins adsorb from your bodily fluids to that implant that's been now inserted in you. So if you know that a certain protein promotes bone growth, then figure out what the energy of that protein is and make your surface match that energy. Similarly for bacteria, if you know that mucin, which is a protein in your saliva, can reduce bacteria growth, then figure out what the energy of mucin is, make your surface through nano features match that energy, and then you can increase the adsorption of mucin, which will inhibit bacteria from attaching. And again, this is all without antibiotics, all without drugs. It's changing the surface to give you the energy you need to promote the adsorption of proteins that these cells depend on to attach or not. So what's pictured on the right side of this slide is that exact point. You have a substrate or an implant. You again have changed that surface energy by putting nanoscale features on it. That can alter adsorption of a protein. One protein that is excellent at increasing bone growth and decreasing bacteria attachment is fibronectin. So that's why I included fibronectin in this diagram. And then you can see a mammalian cell, a bone-forming cell that is attaching to that protein which has adsorbed. So again, the deeper understanding of why these surfaces, whether it's an insect wing or an implant that has nano features, a deeper understanding of how they control cell function is they change in energy, which results in the change of a protein adsorbing and therefore change in cell attachment and function. So we're engineers in this area. You might know engineers like equations. We like to model things. We like to predict things. So we've created an equation around this whole process. I believe we're the first group to actually provide a predictive equation that will tell you, if you give us a sample, our equation will tell you what size of nano feature should you put on that implant or that chemistry to reduce bacteria attachment. Again, without using a drug or an antibiotic. So let me quickly explain this equation, because we love it when other people use our equation and you test to see, does it work for your example? The more data we get on our equation, the more we can perfect our equation. So our equation, again, over here on the left part of the slide, involves three key terms. The first term, pictured on the E sub S diagram, is the energy of the protein that you want to adsorb. So again, if it's mucin, because mucin reduces bacteria attachment, then the energy of mucin is what you would put in E sub S. The next term, E sub 0, comma S, is the starting energy of your material. So this is the flat version of your material. On the slide, A is a flat version of a polymer that is used in many medical devices. That polymer is called polycarbonate urethane. When we use it as catheters or in other implant applications, today it has no nano features, so that's why in A, that sample looks pretty smooth. It's smooth at the nanoscale. So then we go back to our equation. We know what E sub S is. That's the energy of the protein we want to adsorb, like mucin. We know what E sub 0, comma S is. That's the starting surface energy of polyurethane. We can calculate that using contact angles or many other scientific methods. What we then solve for is that REFF. What that means is effective surface roughness. So that effective surface roughness is expanded in the equation below, and it's written for the Z height. So that letter Z in there, we can calculate it, and that tells us how tall our nanoscale feature should be. I could have also written this in the X direction. So X is how wide our nano feature should be. Or I could have written this in the Y direction. How long should that nano feature be? So we back calculate out using this equation, what's the height, the width, the length of that nanoscale feature to give us the energy to adsorb mucin. And that's exactly what's shown on this slide, going from A, the smooth polycarbonate urethane, to B, which has a little bit, couple more nanoscale features, going to C, has a lot more nanoscale features, and then to D, which has the most nanoscale features. We created nanoscale features here by putting carbon nanotubes underneath the polymer. Carbon nanotubes are nanoscale orientations of carbon, and that provides this type of roughness. The more we add, the more roughness we get. And that's what's shown in the graph on E, basically going from 0 weight percent carbon nanotubes in polyurethane, which is letter A, to 13 weight percent, which is letter B, to 20 weight percent, which is letter C, and then to 33 weight percent is letter D. So of course we see an increase of roughness the more carbon nanotubes we add. If you then look at the graph in F, that shows you the change of contact angle, or the change of energy you're getting when you increase nanoscale features. So simply going from, again, 0 to 33 weight percent carbon nanotube going from the image in A to the image in D, we get a more hydrophilic surface. That water drop is spreading more when we have more nanoscale features. We stopped at 33 weight percent, and you might ask why? Why did we stop at the nanoscale roughness we got when we added 33 weight percent of carbon nanotubes to polycarbonate urethane? The reason is the energy we got, as you can see in the graph again on F, the energy we got when using 33 weight percent is the same energy as mucin. So it gave us the same energy to enhance the absorption of a protein, mucin, that reduces bacteria attachment. So, and that's what's shown in G. It basically is an ELISA test, if you're familiar with that, to measure how much of a protein is absorbing to our surface. And we get the most mucin absorbing when we use 33 weight percent carbon nanotubes. So again, what I want to highlight in this presentation is this idea of getting a material off the shelf, adding nanoscale features, and again, we'll talk about other ways you can do this, but adding those nanoscale features to give you the energy to absorb a protein that will then reduce infection or reduce bacteria attachment. I also want to add that we have taken this idea to the FDA, and the FDA has approved our materials which increase in nanoscale roughness for medical applications. Many people often wonder, where is the FDA in terms of thinking about nanotechnology and nanomedicine? And I'm here to tell you there are a number of materials that have nanoscale features that have been approved by the FDA for implantation. And the reason it's occurred so quickly is because we're not changing chemistry, we're changing roughness. If you introduce a new chemistry to the body, your regulatory approval is going to be much longer. If you simply take existing chemistries that are approved by the FDA and put nanoscale features on them, you can have a much quicker path to FDA approval. So some of these materials that I'm going to talk about in part one of my presentation are materials that have been approved by the FDA for implantation, and they use nanotechnology and nanostructured features. So if you ever hear somebody say that there's no real value that has come out of nanotechnology in medicine, they're wrong, because there are a number of materials that have been approved, they're implanted in humans today that use nanomaterials, and they're showing decreased infection, as we're talking about, without drugs. So let me give you some of those examples now. One of the first materials we studied, and this was probably about 6 years ago, was a material called silicon nitride. It's a ceramic. It's commercialized by a company called Amedica, and it essentially is pictured here in the upper right. And I call it smooth silicon nitride because at this scale bar of a scanning electron micrograph, we don't see a lot of nanoscale features. You do see some grooved lines and some micron topography, but certainly there's not a lot of nano topography. So we wanted to reduce infection of this chemistry. So we followed our equation. Again, we plugged in what's the starting surface energy of this silicon nitride sample, what's the protein we want to absorb that can both increase bone growth and decrease bacteria growth. We calculated out the Z height, the X width, the Y length, and the nano rough silicon nitride, the image here in the upper left, is what we got. So this is the exact dimensions of nanoscale features that our equation told us we should create to reduce infection. We also compared this to titanium. Titanium, of course, as I mentioned, is a widely popular material in orthopedics. And we also compared it to a polymer called PEEK, or polyether ether ketone. Polyether ether ketone is the #1 implanted spinal implant material. So it is great mechanical properties. It has a really nice behavior for mechanical situations in the spine. Unfortunately, there's a lot of infection that occurs for peak because it is not it does not resist bacteria growth. So let me show you what we did. We communicated with the FDA. What kind of study should we do to look at infection of these materials? And we got feedback that if we put bacteria on these samples ahead of time and then implanted the samples with bacteria into what's called the calvaria of skulls, that would be a great infection model. So we used rats in this study. Again, each of these micrographs are histology stains in which we had the implant with bacteria on it and inserted that into the skulls of rats. On all four of these images, if you see yellow, that means an infection. That's a stain for bacteria and biofilm-producing bacteria. We also have a stain for bone growth, and that's purple. So if we look at the worst material, again, peak, that I mentioned in the lower left corner of this slide, there was a ton of infection. So the bacteria that we initially seeded grew very fast. They produced a lot of infection. That infection kept bone from growing, as you might imagine. In fact, if you have bacteria in bone, it becomes a really bad clinical situation where you have to drill out. You have to remove the implant and you have to drill out the bone that has the bacteria, because right now there's no way to just kill that bacteria and leave the rest of the bone present. So we quantified this. You can see for peak, 95% of the surface was covered with bacteria after 3 months. Titanium, which is pictured just above peak, did a little bit better, but you can still see a lot of yellow, particularly on the right side of this particular slide. So that yellow, again, signs of bacteria. When we quantified it, we got on average 67% of the titanium surface was covered in bacteria. When we moved to silicon nitride, changing chemistry already improved our ability to inhibit bacteria. So on the bottom right corner, you can see silicon nitride that's smooth. So this was the starting material made by Amedica. Immediately, there was a reduction in bacteria growth. So there's something unique about the silicon nitride chemistry, the silicon nitride surface energy that reduces bacteria attachment and growth. But still, we saw 10% of the surface of silicon nitride, of the smooth silicon nitride, covered with bacteria. So 10% is still a pretty large amount. In fact, you can see there's gaps in bone growth surrounding that implant. So those gaps in purple color, the white color, are regions of no bone growth. And that's because there was bacteria that was present in those locations. So there's still reason for improvement for that smooth silicon nitride. So then you look at the best material, and that was the silicon nitride that had the nanoscale features, the nano rough silicon nitride. And again, no antibiotics were used in this study. These are simply the same chemistry, silicon nitride, that had those nanoscale features. We ended up with 0. No bacteria present on that surface after 3 months. That, of course, allowed for a lot more bone growth. In fact, the most bone growth out of these 4 samples, and it was at about 41% of that surface of silicon nitride was covered with bone. Let me give you another example. So now instead of silicon nitride, think of titanium and how we can improve upon titanium and reduce the infection of titanium-based orthopedic implants. Another way you can create nanoscale features is by a process called anodization. And that's what's diagrammed on this slide. Anodization is a real simple, well-understood process where you put titanium, serves as the anode, into an electrolyte solution. We use HF, or hydrofluoric acid. You apply a voltage for a certain period of time, as indicated on this slide, and you can create nanoscale features on bone pins. So this slide shows you a low magnification image of this anodized bone pin and a high magnification picture showing nanoscale features of this bone pin. Again, bone pins are used all the time in orthopedics for various bone fractures. In this study, we were very interested in creating a better device for an amputee. And an amputee is one of the most difficult orthopedic situations to find solutions for. As you might realize, whenever you lose part of your limb, there are several strategies to try to return function to that limb. One is simply to fit a plastic, prosthetic, or the remaining limb around what's left. Or in this case, a much better strategy is what's called an osseo-integrated limb. This is where you take the screw, you put it into the remaining part of the limb, and then you fit a plastic prosthetic around the screw. This is to increase stability and to decrease abrasion and friction that is felt if you don't use a bone pin and you just put that plastic prosthetic around the remaining limb. So nonetheless, we took some pins that, again, are inserted into the remaining limb of an animal model we have for an amputee. Some of those pins were anodized to have these nanoscale features that you can see on this slide. Some of them were not anodized, so they were nano-smooth features. We, of course, followed our equation, as I mentioned before, to tell us what size nanoscale features should we put on the pin to give us the energy to absorb proteins necessary to reduce infection for these devices. Here's an X-ray of our amputee rat model, and again, you can see the femur right here that still exists inside of the rat, and the amputee model resected the rest of the limb, and there's that bone pin that goes right into the remaining limb. So again, the reason why this is so complicated for orthopedic surgeons, for people developing improved biomaterials, you have a metal that's sticking out of the body that this prosthetic will fit on. So that metal has to bond to bone, it has to osseointegrate, but it also has to bond to soft tissue and skin because you need the wound to close around this screw to keep bacteria out, to minimize inflammation. And just to highlight how important of a problem this is, there was a recent survey that was done by the Department of Defense in the United States. This survey found that more people were unhappy with their prosthetic. If they were an amputee, they were more unhappy with their prosthetic than if they received no prosthetic at all. So in other words, as orthopedic surgeons, as biomaterial specialists, we try to create a better remaining limb for amputees, but the data is showing, the survey is showing that our soldiers were much more disappointed with receiving these kinds of surgeries than if we did nothing at all. So that really highlights this problem we have with amputee biomaterials. So the results from this study were incredibly promising. I don't have a movie here to show you, but I do have the rat. Qualitatively, you can see where the screw is in the back right leg of this rat. And what we saw was the rat was walking on the anodized, the nano-textured titanium implant 3 days after surgery. For the control pin, the one that's used today, this rat did not walk on it for 28 days. So, and that's as far as we took this study, was for 28 days. So clearly, the rat felt more comfortable with this anodized implant than unanodized or nano-smooth implant. When we look at the wound, the exit wound, where the metal is coming out of the rat, again, the spot that the prosthetic would fit on, it's clear to see why the nano-structured implant did so much better. This first row of images is the pin that's used today. You can see an open wound. After 14 days, 21 days, 28 days, that red tissue that you see is inflamed tissue. It got infected. There were signs of bacteria on the screw crawling into the femur into the rat. So it was a clear problem when not having nanoscale features. The middle row and the bottom row are two examples of nanotextured surfaces. One was that nanotubular surface geometry that we created using anodization. If we change the process a little bit, we get what's called nanorough, not nanotubes, but just hemispheres of nanoscale features. Either type of nanoscale feature we created, you can see the wound closed. So the skin grew onto the screw after 14, 21, and 28 days. This again kept bacteria out. It reduced inflammation, and it allowed the pin to grow into bone. So again, no antibiotics were used. This is simply putting nanotexturing on these devices, which helped reduce bacteria growth, allowed for skin to grow, allowed for bone to grow, and allowed for a successful material. So this one is also approved by the FDA. The silicon nitride example, I did not mention this at the time, but that has also been approved, the nanotextured version, by the FDA for orthopedic applications. So here's two examples of nanotexturing that has reduced infection and has been approved by the FDA for implantation. Let me give you a last example of a nanotextured surface before moving into the next part of my talk. So you might wonder, okay, this approach of nanotexturing seems to work on metals, which is what titanium is. It worked on silicon nitride, which is a ceramic. So how about polymers? Can you create polymers to have nanotextures to reduce bacteria infection without using an antibiotic? And, of course, I'm going to say the answer is yes. And you can see here the third example that I'll give is for catheters or endotracheal tubes. These are polymeric materials, clearly. And again, our objective here was to create nanotextures on the inside of these catheters or endotracheal tubes to reduce infection. This is also a very big problem in medicine. If you have a catheter for a long period of time, of course, the chances of getting infection go higher and higher, and many times the clinician does not know you have an infection until they see bacteria in your blood, a systemic infection. And this clearly then allows the bacteria to go everywhere in your body. So we need a much better approach to keep bacteria from growing on these catheters, these endotracheal tubes, to reduce systemic infections. So our process that we followed to create nanostructured features on these polymers was very straightforward. Catheters are very inexpensive. So in order for this to be adaptable to the clinical community, we needed a very inexpensive process to create nanoscale features. Catheters are, $1 or $2, so you cannot propose something very sophisticated that would cost thousands of dollars to create nanoscale features on catheters and expect them to be adopted by the industry. So we used a cast mold technique. Very cheap, very simple. And again, our material we used to copy roughness onto the polymer was our anodized titanium, the same material that I just discussed for that amputee model. We followed an anodization process. We were able to generate nanoscale features similar to what I showed you before. Here's a picture in the upper right of these unique nanoscale features we create on titanium. That roughness can then be copied over to a polymer by simply pouring a mold, making a mold of this titanium, and then pouring polymer like PDMS, which is polydimethylsiloxane, into that mold. And you can copy over the roughness just like any kind of cast mold process that's used in many different industries. And here you see in A is the starting titanium, the nano smooth titanium. The picture right below it is a PDMS copy of that starting titanium. So both are very smooth at the nanoscale. If you then look at B, again, that's our anodized titanium, has all those nice anodized nanotubes, and our PDMS copy of that titanium is in picture D. So we were able to copy the roughness over to the polymer. Again, we followed our equation. What size of features do we need to get the energy to allow for proteins to absorb to this catheter that would reduce an infection? We measured wettability. Remember, our goal is to transform that surface energy to one that would promote protein absorption. So here you can see in A, our starting PDMS. So this is the catheter you would find today on the market. And then in B is a nano-textured one. Clearly you see a different spreading of that contact angle, again proving you can change wettability. You can change surface energy by simply employing nano-textured features. We also wanted to check chemistry. So without going into too much detail, we used XPS, X-ray photoelectron spectroscopy. We saw the same exact chemistry between the starting PDMS and the PDMS that had nanoscale features. This, again, important for the FDA to show similar chemistry, just changes in nanoscale feature. Bacteria results showed exactly what we expected. So here is for Staphylococcus aureus, a bacteria that commonly infects catheters. In fact, you might be aware of MRSA, which is the antibiotic-resistant version of this bacteria. And you can see after 24 hours, 48 hours, the density of bacteria or the colony-forming units per ml significantly lower when we simply put nanoscale features on those implants. Nanoscale features resembling those that are on the insect wing that I presented in the beginning. Just to highlight the mechanism, in case I haven't been clear about this, but here we also noticed that there was another protein involved, not just mucin, which can really help reduce infection on orthopedic implants or dental implants, but there's another protein called casein that you have in your body. And again, you look up that energy, create these catheter surfaces to have nanoscale features to match the absorption of casein, and that's why we're reducing bacteria growth. So the mechanism of action, unique wettability, increased protein absorption like casein. Casein inhibits bacteria attachment and growth. So that's the mechanism we believe occurred. So let me switch a little bit into another part of nanotechnology that has been receiving a lot of attention in the literature, in the medical community. Again, the first part was all about nanotexturing of implants. And there are many, many examples. I could have probably spent 2 days talking about other implants that have nanotextured features that are approved by the FDA and are now in humans. And they're showing improved tissue growth, reduced infection, in some cases reduced scar tissue formation, all of those things showing much promise that without drugs, you can control cell behavior using nanotextures. Another area, though, is nanoparticles. Nanoparticles have been a little bit harder to implement in medicine and get FDA approval, and there's many reasons why. But let me talk about the pros and the cons of nanoparticles. Many people are excited about nanoparticles because they are now very small and they can get inside of cells. They can get inside of tissue-forming cells. They can get inside of bacteria, and they can actually turn on and off individual parts of cells. So you could create a nanoparticle to go into a bone cell nucleus and get involved in DNA, attach to it, disrupt it. You can have nanoparticles go into cancer cells and begin to heat up under controlled infrared excitation, and that heat that they generate could kill the cancer cell. So you can use nanoparticles to probe parts of the body that were once unthinkable with micron particles. So that's the promise. And I'll show you some examples of how we're using nanoparticles for bacteria. But the weakness or the problem of nanoparticles is the same thing as its advantage. If you can now have nanoparticles penetrate your skin, pass through the blood-brain barrier, go into cells, you could potentially have increased toxicity from these particles because they can go in places that micron particles were once unable to. So you have to be very, very careful. And the FDA is right on top of this, asking for a lot of studies if you're proposing a nanoparticle to treat a particular disease. But here you can see, again, a diagram of a cell. You could think of this as a cancer cell or even a bacteria, where another great advantage of using nanoparticles is through their small size and increased surface area, you can now generate more of what's called reactive oxygen species than you can with a larger micron particle, right? Envision a box, and that box has micron particles for one box. For another box, it has nanoparticles. Same volume. The nanoparticles have much more surface area, which means if they produce reactive oxygen species, they can produce a lot more reactive oxygen species because they have more surface area. Reactive oxygen species, as you might know, can kill bacteria. So it's a very common way to kill bacteria. Many drugs are involved in reactive oxygen species, but we have seen without drugs, nanoparticles can result in the generation of reactive oxygen species to kill those bacteria. So there's a lot of advances going on with silver nanoparticles that generate reactive oxygen species, things like ceria oxide. That is a great modulator of reactive oxygen species. So there's a lot of really fascinating nanoparticles that now can be used to kill bacteria that before we wouldn't even think of using those kinds of chemistries. So here's an example of another fascinating advancement in using nanoparticles in medicine. So many times when you have an infection, you might be going through cancer treatment. In fact, infection rates for cancer treatments are incredibly high, and that's clearly because patients are going through chemotherapy. They have weakened immune systems, which make them more susceptible to bacteria and infections. Well, we are creating, and other people in the field are too, what we call synthetic immune nanoparticles. So it's a boost to your immune system. These nanoparticles, you can see dozens of them in the picture on the left. There's about 4 or 5 of them on the picture on the right, the higher magnification. We functionalize these synthetic nanoparticles to attack bacteria. So we know certain receptors on the bacteria. We put those complementary attachment regions to our nanoparticles so that our nanoparticles attack that bacteria. This bacteria on these slides are E. coli. E. coli clearly is in a lot of food and can cause problems for people if E. coli gets inside of you. So what these particles do is they first attach to the cell membrane. We have seen many nanoparticles can kill bacteria simply by attaching to their membrane because they clog the membrane. They keep nutrients from going in because they've clogged the membrane, and they keep waste from coming out, again, because they've clogged the membrane. So clogging the membrane with a nanoparticle is a great way to kill a bacteria without a drug. But moreover, some of these particles, and you can see this in the right image, can actually go into the bacteria. So one of these particles has silver nanoparticles inside of it, and it passes the cell membrane and then can release some of these other nanoparticles to generate reactive oxygen species to then kill the bacteria. So the other value in this synthetic immune nanoparticle is we can also use it as a detecting source. So can it tell us that that bacteria is present in your body? Not only kill it if it's there, but tell you if it's there. And we are creating these nanoparticles to be sensors in your body. And how this works is if you look at this picture on the left with the dozens of nanoparticles surrounding the bacteria, once they do that, if you have a conductive material inside the nanoparticle, then it increases the conductivity in that region, which you can detect using some kind of imaging modality. So because you put a conductive material in there, or maybe in some of our studies we put iron in there, once they congregate around a bacteria, you can image that and you can determine that in fact you do have E. coli or some other bacteria that's present. So we're pioneering this field that's called bacteria theranostics, which theranostics means same material does the therapy and same material does the diagnostics. In contrast today, where we have one process that does the diagnostics and one process that does the therapy, we're creating things like these synthetic immune nanoparticles to do both at once. So speaking of iron oxide, there's also approaches you can use nanoparticles to kill bacteria after a biofilm has formed. So once bacteria attach to a surface or a tissue in your body, if they grow to confluence, if they cover the surface and then they form multi-layers, they'll eventually form what's called a biofilm. Biofilm is kind of this mucus, sticky substance that is impenetrable to antibiotics. So it's kind of a shell around the bacteria that creates a little microenvironment that keeps antibiotics from going in, and this is why antibiotics have been useless in fighting bacteria once you have a biofilm that forms. Well, although the antibiotics cannot penetrate the biofilm, nanoparticles can. And in one particular area of study that we have, we're using iron oxide nanoparticles, or some people call them polymersomes, which are magnetic. Iron's a magnetic chemistry, so you can basically use magnetic power, magnetic force, to get those nanoparticles to go through the biofilm, then go into the bacteria, disrupt the biofilm, kill the bacteria, whereas at one point you couldn't treat it, you now can with antibiotics. With iron oxide nanoparticles. Antibiotics were not effective. So here's a picture of some of those collection of iron oxide nanoparticles. Below, you can see a solution of them. But again, what makes them beautiful is they're magnetic. So once they're in the body, you can control where they go. So if you have an infection in a remote, hard to get to location in your small intestine, you could inject a patient with these iron oxide nanoparticles, use a magnetic force to drive those particles to that infection, determine if you have an infection, because again, you can image these iron oxide nanoparticles, and then in real time, you can watch. Is that infection reducing? Is it ineffective, your treatment, and you need another injection of iron oxide nanoparticles? You can watch the treatment occur while you're in the hospital or in that doctor's suite. Here's a quick experiment to show you how well a magnet does in terms of these iron oxide nanoparticles. These two images are the same field of view. One is a bright field image where you can see the dark black area is a magnet. So we put a magnet underneath a petri dish, we add all these bacteria, grow a biofilm, and you can see, in the bright field image where that magnet is situated. Again, the same image, just under fluorescent mode, is on the right. And this is what's called a live dead stain. So we apply our magnetic nanoparticles and then we stain for green, means that the bacteria are living. Red means the bacteria are dead. So you can see right where we place the magnet, that's drawing the iron oxide nanoparticles into the biofilm, into the bacteria, and it's killing them. Where we don't have the magnet, we're still seeing live bacteria. So again, proof of principle that you can use a magnet to drive these nanoparticles inside a biofilm to kill bacteria. Another chemistry that is getting a lot of attention today is called selenium. Selenium is part of our diet. You find it in many foods. In fact, where this interest in selenium came from is there have been studies to correlate if you do not have the daily recommended nutritional value of selenium in your diet, you're more likely to get cancer than if you have the nutritional diet of selenium. So there's a thought that selenium might be involved in anti-cancer properties. In fact, there are now containers of selenium, vitamins of selenium that you can purchase. Well, there's many similarities between killing cancer and killing bacteria. So we've been very interested in taking that wonderful information on how selenium is being used to kill cancer cells, kill tumors, and kill bacteria. And just to make a long story short, selenium is often proposed to kill cancer cells and bacteria, as we have shown, since it interferes with bacteria that are proteins that are inside bacteria. So as soon as selenium crosses the membrane, it dissolves. The selenium ions interfere with some proteins in bacteria and disrupt their function, which results in death of the bacteria. Same thing for a cancer cell. The selenium gets inside the cancer cell, interferes with some bacteria, with some proteins that are in cancer cells, and that kills the cancer cell. So we were one of the first groups to develop a chemical reaction that can coat almost anything with selenium nanoparticles. So we're doing in the body applications, outside the body applications. Bacteria are everywhere. So we're coating a lot of surfaces with selenium. This slide shows you how we took an uncoated paper towel in B and coated it with selenium nanoparticles. So all of those little white dots in A are selenium nanoparticles. It's a simple chemical reaction. We actually have a spray. We can spray computer screens, doorknobs, you name it. We can spray this selenium nanoparticles and they stick to these surfaces. So when doing so, we're seeing great decrease in bacteria attachment, decrease in bacteria growth. Here's just some of the raw data where this is for, again, Staphylococcus aureus, but we've seen this for many bacteria, where you see significantly reduced 5 log reduction in bacteria growth after 24 hours, 48 hours, 72 hours when coating these paper towels with selenium. So it might sound trivial, but paper towels are a source of bacteria contamination. Think of a hospital. In the hospital, you wash your hands after, you're in the restroom and you throw away the paper towel. Well, there is now bacteria on that paper towel in the trash can in the hospital. That's a source of bacteria contamination that, if not treated appropriately, could end up affecting health of people. So what we propose to do is to treat paper towels with selenium, nanoparticles of selenium, to really keep bacteria from growing on these paper towels or on other materials that might be in the hospital or everyday life setting. We've also been able to coat metals, stainless steel, ultra-high molecular weight polyethylene. You can see on this slide, going from the left to the right, uncoated versions of these metals and polymers, low dose of selenium. You start seeing a couple of white dots, medium dose, high dose. You see more whites, again, very surfaces, and they're effective at reducing bacteria attachment. We've also seen them effective at reducing virus attachment. So things like Zika, Ebola, the flu virus, these selenium nanoparticles can keep those entities from absorbing, which again would reduce transmission from touch to touch on these surfaces. Here's a great picture that describes it all too. We've done this for polyvinyl chloride. PVC is a common polymer used for catheters. You can see on the uncoated polyvinyl chloride catheter, millions of bacteria, and these are Staphylococcus aureus, again, have attached to this PVC. The simple coating of selenium on there significantly reduces bacteria attachment and growth. And again, this is not using an antibiotic. So that moves me to the last part of my presentation, which is, again, a very active area of research in the nanotechnology space. So we talked about nanotexturing, nanoparticles. This is self-assembled nanomaterials. These are injectables. You would inject these into the body where you think you might have an infection, or you apply them onto the surface of skin in which you think you might have bacteria presence. Then these materials assemble in a controllable way to selectively kill bacteria. So this is the field of self-assembled nanomaterials, which is growing and growing in popularity because it's sort of like a pre-programmed nanomaterial. You know it will self-assemble into a controllable way to kill bacteria. Our version of self-assembled materials we're using to fight bacteria are called antimicrobial peptides. So what we do is we simply take things like hydrophobic amino acids that can attach to the membrane of a bacteria. We then add a group that's hydrophobic, add a group that's hydrophilic so that it will self-assemble into a rod to then interject into the bacteria membrane, kind of stab the bacteria membrane and cause the inside to come out. What is nice about these self-assembled antimicrobial peptides is they are very strong interaction with a bacteria cell membrane, as you can see on the left, but they have a weak interaction with mammalian cell membranes. And really the reason for that is mammalian cells, which we don't want to kill, have things like cholesterol, they have other proteins that are present in the cell wall, which won't be stabbed or won't be influenced by these self-assembled chemistries. So they're really selective towards killing bacteria, which is very, very important. So here's a visual description of what these molecules do. Again, we design them to self-assemble into rods. They come in close contact with the bacteria membrane, as pictured here, and then they start going into the bacteria membrane. Once they go through, part of it goes through the bacteria membrane, part of it is inside the cell. That generates a hole, and from that hole, everything from inside the bacteria comes out, and it dies. So these structures, the ability of these self-assembled materials to assemble into these fibers, nanofibers, is very important because you need something long enough to go through the membrane, and you need something small enough that it doesn't come out the other end, but it goes right into that cell body, the bacteria body. Here's the specifics of a molecule, that one of the molecules of many that we're studying. And again, we need a hydrophobic tail group. We need a beta sheet forming domain, a charged domain, and then a functional domain. The hydrocarbon tail group helps with self-assembly in a fiber-like formation. The beta sheet also helps in that self-assembly, and the functional domain is what allows us to target E. coli or Staphylococcus aureus or Pseudomonas aeruginosa. So we can use that red region to target a specific bacteria. So again, the picture of how this happens, we create monomers of these cationic self-assembled peptides. They self-assemble into cylindrical nanoparticles or rods. They first attach to the cell membrane, but then because of their charge, they go into the cell, creating a hole, creating this leakage of the cytoplasm, which results in their death. But please keep in mind, the same thing does not happen for the mammalian cells because of the inability of these particles, these fibers, to get through a mammalian cell membrane. So again, a diagram of this, the specific chemistry we're looking at, if you're interested in that, the hydrocarbon group, this beta forming sheet, and then the functional group there in orange, and how it looks, if we diagram this in terms of a chemical structure. So what I want to highlight is just a real quick study that we did with these molecules, in which we highlighted the reason why they need to form nanostructures. So nanotechnology is all about the importance of the nano size scale. So what we have done is a series of studies that show how important it is that these materials assemble into a nanofiber as compared to a micron fiber or a nanosphere. the nanofiber is very, very important. So we created our material step by step, and we added it to bacteria. So the first step was just this bicarbon peptide. This orange region, as I mentioned before, is the peptide that targets the cell membrane, the bacteria cell membrane. So we use that as one material. Another material we call CVK-PA. This is really the beta sheet self-assembly part. So we added that just to the peptide. That was our second material. Our third material is the whole entire thing. So we now have the hydrocarbon backbone, the beta sheet formation, and that region that attaches to the bacteria membrane. And as you'll see, it's all three of these together, the ACA-PA, that works. If you remove any part of that, this three-component system, if you remove any part, the whole thing doesn't work and it doesn't kill bacteria. We, of course, determined the critical micelle concentration. So this is the critical concentration at which each of these components would kill bacteria. And you can see just the bicarbon, just the peptide that attaches to the membrane doesn't kill bacteria at all. Under the critical micelle concentration, there's Na, not applicable. It doesn't kill it. The self-assembly region with the beta sheet formation and the bicarbon and the hydrophobic backbone had a critical micelle concentration of around 50, which means that's the amount necessary to kill the bacteria. Here's what these structures look like. Again, you can see these nanofiber formations. Of course, if you increase the concentration of these self-assembled materials, you'll get longer and longer chains. So we need to have the right concentration that will penetrate that bacteria wall. We confirmed secondary structure to show that each one of these three materials is the structure we think it is. And most importantly, what I want to share with you is the ability of these materials to reduce bacteria growth. So we looked at gram-positive bacteria, MRSA. You can see with time, we're able to inhibit bacteria growth. With gram-negative bacteria, this is MDR, E. coli, or multi-drug resistant E. coli, we completely eliminated using these self-assembled molecules. Live dead stains, similar to what I described before. Green means alive, red means dead for MRSA, the horrible drug-resistant Staphylococcus aureus. This self-assembled molecule almost completely inhibited MRSA growth. Multi-drug resistant E. coli, again, you can see high efficacy at killing those bacteria. But here's the part that I mentioned before. You need each part of this self-assembled molecule to work. So on this slide are the building blocks by themselves, not the collective molecule, but just each building block. The bicarbon peptide, the peptide amphiphil, they don't work. The y-axis here is the number of colony-forming units. It's the same, no matter what concentration you use. So only when you have the combination of the beta-forming sheet, the bicarbon peptide, and the hydrophobic backbone do you get that fiber to then kill the bacteria. So that's a real definition of nanotechnology. It has to form a nanostructure. We've also looked at the mechanism of action. For those of you that are interested in bacteria, lipopolysaccharides are a major component of the membrane of gram-negative bacteria. And you can see here the red is our self-assembled molecule. That is what attaches the most to that LPS region of the bacteria membrane. If we just had the building blocks, which are in black and blue, we don't get a lot of attachment. And this last slide, before I summarize, is so important because it really shows you what's happening to the bacteria. So the nanorods that we created, you can see when we expose them to MRSA or E. coli, the membrane is getting disrupted and the cell is basically, internal components are coming out of the bacteria. So it's clearly dying as soon as we disrupt that cell membrane. Whereas the controls, you see the cell membrane is intact. So I guess the very last thing that I'll show here is cytotoxicity. So one important part is we have identified a concentration at which these self-assembled materials work for bacteria, but they do not work, and they do not kill human dermal fibroblasts or healthy cells that we don't want to kill. So they are selective towards bacteria. And we can charge them. If you charge these self-assembled materials, they'll coat surfaces. So tying this back into the beginning of my presentation, you can actually use these on medical devices, coat them on medical devices by inducing a charge, and that will help contact kill bacteria. So I hope I shared with you a couple of future directions, current directions in nanotechnology. I showed you some current FDA-approved processes that we call nanotexturing or nanostructured surfaces, which are eliminating infection without using antibiotics. I also provided you a couple of examples of nanoparticles, a little more risky, a little more work involved in determining biodistribution and other things. And I ended the presentation with a really futuristic direction of self-assembled nanomaterials, where you can now have a chemistry that can poke and prod and disrupt a bacteria membrane to cause it to die. All three of these areas are really, really exciting intersections between nanotechnology and eliminating bacteria growth without resorting to drugs. But let me leave you with one final thought. And that final thought is, in all of these approaches, most likely you would still have to determine if someone has bacteria in them and then give them a solution. I gave you a small example of an immune cell sensor that we're developing. Here's another sensor that we're developing. And the purpose of these sensors, which I'm convinced will be the future of medicine, the promise of these sensors is that they, in fact, can determine if bacteria are present surrounding your implant, and then, if so, kill them. We don't have that technology now. We don't know in real time if this is occurring after you get an implant. So quickly, what we're doing is we're taking a hip implant, we're growing carbon nanotubes, that's what's pictured in the bottom right, off of this surface. Carbon nanotubes can measure the resistance and therefore determine if a bacteria is attaching to the implant. Bacteria have a different resistance than bone, which is different than scar tissue. So we can identify if bacteria are on your implant in real time. That information is then sent to a computer or your iPhone to tell you that you have bacteria surrounding some of your implant. We've already made an appointment for you with your doctor. So it's an intelligent system that communicates and talks to you about what's happening inside your body. The third part to this sensor is that we developed a polymer that can release a nanoparticle or a self-assembled material on demand. And it does so because it's a conductive polymer that we apply an electromagnetic field. It will degrade and release whatever's inside of it. So we now have the ability to determine, is a bacteria present? Communicate to a handheld device to your clinician, yes, it is present. And then thirdly, release something, nanoparticle, self-assembled material, to kill the bacteria if it's present. This is a true intelligent material that has to be the future of our research in medicine. So I'd like to thank you for paying attention. Here's some acknowledgments for funding sources, of course, that have occurred throughout my career, which has enabled us to do this. And of course, my lab group has enabled me to do all this research. I encourage you, go to our website, learn more about the things we're doing. Nanotechnology and nanomedicine is a fascinating area of research that I hope more people get involved in. Thank you.